Hello, my name is Tess Cherlin and I'm a PhD student at Thomas Jefferson University in the Genetics, Genomics, and Cancer Biology program. I'm going to talk to you today about the snowball effect, how lots of small changes in our cells can lead to cancer. So, as I said, cancer is a snowball effect. It begins with one screw coming unloose, and then more screws and nails and bolts, and they all start coming undone. And then more momentum and more until finally one small problem leads to a large problem and the entire house comes crashing down into a chaotic heap. But what if we could fix or find that first screw? When my sister's friend's dad was diagnosed with late stage cancer, it was really sad. I also remember thinking, what if they had found it sooner? We all know on some level that if we are diagnosed with cancer, the earlier the better for survival. For my PhD work, I am interested in understanding the genetic material in each of us that has the ability to change from healthy to harmful. By observing these changes, I hope to find unique genetic molecules that are cancer detecting. Wouldn't it be amazing if, just like taking a simple pregnancy test or drug test, that we could know relatively instantly if a person we love has cancer? Better yet, if we could find out early enough to help save them from long treatments, painful therapies, and death. Today, I hope to tell you about genetic material called microRNAs and how they are turned off in cells. Then I will explain how this process changes when in a cancerous state. Finally, I will discuss how identifying these microRNAs and these changes of microRNAs can lead to predictive technologies called biomarkers. In our bodies, there are millions and millions of cells. To be exact, we have around 37.5 trillion cells. Each one of those cells is required in order to keep us healthy. If we think of a cell as a house, we could categorize the stuff in that house into structural and functional. For example, our walls are structural and they hold the shape of the house, while a security system is functional. It's surveying everything and making sure nothing's going wrong. Cells, like a house, require both structural and functional materials called proteins. Some proteins are walls and some proteins are the security systems. The functional proteins can turn on and off other proteins which are required to do necessary things, both structural and functional. But functional proteins can turn on functional genetic material called microRNAs as well. Just like how a security system might signal for an electrician to fix the refrigerator from getting turned off, so too can microRNAs be turned on or expressed by functional proteins to modulate the amount of other proteins that exist in a cell. microRNAs, therefore, are there to keep a cell balanced and happy. And these microRNAs are functional molecules that are turned on by functional proteins. But what if our hypothetical house gets a glitch in this functional security system and begins to malfunction? In a cell, when a protein malfunctions, it's called a mutation. In this house scenario, the mutation or glitchy security system turns on functional microRNAs that they're not supposed to. And these unintentional microRNAs are going to go all around the house and start shutting off things inappropriately. The first functional protein situation that these microRNAs decide to shut off are going to be the lights. While having no lights is inconvenient, it's not really a huge problem because you could just call a person to come and fix the lights. But the first mutation, while it's inconsequential might lead to microRNAs going and shutting off other things, leading to other problems. And eventually mutations will mount and grow until you have the windows disappear, or the walls come down, or you have no running water. All sorts of problems can happen until you finally end up with a house that doesn't structurally resemble or functionally act like a normal house anymore. Just like this house, a cell can acquire so many mutations that it doesn't look like or function like a normal cell anymore. The cell will begin to grow out of control and become a cancerous tumor. When this happens, you can't fix all the little problems that you began with. You have to treat the big one. You have to either cut the cell out and remove it, or try and kill it. A lot of cancer therapy is focused on figuring out ways to kill tumors with drugs, immunotherapy, and radiation. But my research focuses on stopping the whole snowball effect before it starts. The original microRNA in our story was turned on inappropriately from the beginning, and we thought that that was probably harmful. However, this might actually be our greatest hope for early detection of cancer. MicroRNAs are special in that they can leave a cell and enter the bloodstream and circulate throughout the body. So, it would be really great if we could take a simple blood test and see if microRNAs that are causing or predicting cancer exist in this blood sample. The research that I do looks specifically at breast cancer cell lines and analyzes all of the microRNAs in these breast cancer cells. Then I do a lot of analysis to try to figure out which microRNAs are cancer specific. Once I do that, we can use these as a technology called a biomarker to identify microRNAs as a predictive tool for cancer. Hopefully the work that I do will lead to microRNA biomarkers being used for early detection in all cancers. Today, I hope you can take away that genetic material called microRNAs are turned off in cells as a part of normal healthy cell function. However, when a person has cancer, microRNA levels change, leading to genes being turned off inappropriately. But by identifying the microRNAs changes in cells, we can make predictive technologies called biomarkers to identify cancer sooner. Thank you so much for your attention and listening and learning about microRNAs. And if you have any follow-up questions, feel free to ask. Also, if you want to learn more about biomarkers and microRNAs, you can check out a really cool TED Talk by Jorge Soto, who designed a microRNA scanner that can be analyzed with a smartphone. It's really cool. Thanks, and have a great day. Bye.